Okay, so for part 3 here, what I'd like to talk to you about, we left off in part 2 talking about differences in gene expression between prokaryotes and eukaryotes, some of the not technical, but some of the procedural differences and system differences between the two. But there are other things that can influence genetic expression. And one of the most common and complex types of influences on genetic expression is the environment itself. What is going on in the environment that is going to cause microorganisms to respond to these environmental stimulus, and those responses require some kind of genetic expression. So in order for prokaryotes to be able to respond, for bacteria to respond to these environmental changes, one of these environmental influences due to what's known as signal transduction. In signal transduction, bacteria are going to receive some kind of signal from outside of the cell, from their environment, and they're going to respond to that environment by translating specific genes in their chromosome. They do this in order to monitor existing conditions. What types of sugars are available? What types of nutrients are available? Do I have the proteins that I need? Do I need to excrete, if there are proteins present, can I take those proteins in, or do I need to excrete some type of protease to break those proteins down? They're going to use those responses in those the monitoring of existing conditions in order to develop the correct response to those conditions. This is no different than it might be chilly outside, so you make sure that you put a sweater on before you go out. You are responding to that external environment by mounting a response of putting on a sweater. An example of this is quorum sensing. Now, quorum sensing is a way that bacteria will talk to each other, and they do this chemically, very much the same way that our own cells talk to each other chemically. Now, in quorum sensing, this was first discovered by one of my scientific heroes, Bonnie Bassler. She is a microbiologist and full-time professor at Princeton University, in charge of her department, that sort of thing. And Bonnie Bassler is the one who discovered the genetics and understood what was happening in the quorum sensing. So she's basically the founder of quorum sensing in bacteria. And she was studying the bacteria that is part of the Pseudomonal bacteria that is part of lung infections in cystic fibrosis patients. And I have a TED talk that she did here that I'm certainly going to show you guys. And she's really quite amazing. So in the quorum sensing here, bacteria, these Pseudomonas bacteria that are colonizing in the lungs of patients who have cystic fibrosis, will actually start forming a biofilm. And we talked about biofilms in Unit 1. So in these biofilms, they start talking to each other and chemical signals going back and forth, and they're basically sensing density of their own population. I'm a Pseudomonas. Are you a Pseudomonas? Yeah, I'm a Pseudomonas. Are you one? And they're talking to each other chemically in this sense. And once the chemical density, the level or population gets high enough, it signals a collective response. And in this sense, all of the Pseudomonas bacteria will sense the same concentration or population number at the same time. This increases to a specific threshold the concentration of the chemical signal that they're sending out. Everybody's saying, I'm a Pseudomonas, kind of like a density level. And all of the cells respond at once by producing a toxin or doing something that causes the patient to go into some kind of respiratory arrest. So this quorum sensing phenomena that Bassler is talking about is due to population density, and it is a concentration-dependent reaction. So I'm going to let Bonnie Bassler show you in this TED Talk the genetics behind how these bacteria talk to each other and why her discovery is so important and what she's working on next.
So now what we understand is that all bacteria can talk to each other. They make chemical words, they recognize those words, and they turn on group behaviors that are only successful when all of the cells participate in unison. And so now we have a fancy name for this. We call it quorum sensing. They vote with these chemical votes, the vote gets counted, and then everybody responds to the vote. And what's important for today's talk is that we know that there are hundreds of behaviors that bacteria carry out in these collective fashions. But the one that's probably the most important to you is virulence. So it's not like a couple bacteria get in you and then they start secreting some toxins. You're enormous. That would have no effect on you. You're huge. But what they do, we now understand, is they get in you, they wait, they start growing, they count themselves with these little molecules, and they recognize when they have the right cell number that if all of the bacteria launch their virulence attack together, they're going to be successful at overcoming an enormous host.
Hi, I'm delighted to be back to give you a little progress about what we've been doing in quorum sensing. And so today I want to tell you one story about how we're taking what we learned about these bacteria talking together and trying to interfere with that conversation to make a fundamentally new kind of antibiotic. And so the story I'll tell you about concerns this pathogen, Pseudomonas aeruginosa. This is the bacterium that kills people who have cystic fibrosis, it kills immune-compromised people, and it causes infections when you get a catheter, a stent, or a breathing tube. And the reason Pseudomonas is so virulent is because of this chemical communication, this quorum sensing. What Pseudomonas does is that as it grows, it makes and releases small molecules, which are the red triangles on this slide. And so as the cells grow, these molecules that are outside of the cells increase in proportion to cell number. And as you heard on the clip, when the bacteria detect that those molecules are there, they interpret that that means there's other cells around. And then as a collective, all of the bacteria together make a biofilm, which is how they sit on surfaces and adhere to tissue, and then the group together secretes the poisons, the toxins that make us sick. So that's quorum sensing. And so we want to be able to interfere with that conversation. And so we know what the molecule is that Pseudomonas talks with. It's the one that's on the left side of this slide. And so what we did using chemistry is we changed the structure of that molecule to make the one that's on the right. And so what that chemistry did was it changed the signal molecule, the word, into an inhibitor. So we changed the molecule that turns on quorum sensing into a molecule that shuts down quorum sensing. So what happens if you have such a molecule? So first I'll talk about biofilms. So in this Petri plate, what we've done is we've put Pseudomonas in the middle of the Petri plate. And what I hope you can see is that the bacteria have spread out to the edges. That's this biofilm formation. As a group, they move out over the plate. And that could be like your tissues. But we have this inhibitor. So now if we do the experiment, we put the Pseudomonas in the plate and we add the inhibitor, what you can see is that the Pseudomonas can't move. So that's good. That's step one in the infection. It seems like our inhibitor can shut down biofilm formation. The next question for us is what about these poisons, these toxins that Pseudomonas secretes? So now you're looking at an experiment, and in the left-hand test tube, that's wild-type Pseudomonas. It's doing quorum sensing and it's secreted these toxins. And when it secretes those toxins, the bacteria turn green. In the middle test tube, that's a mutant that we've made, where we've knocked out its quorum sensing system. So that mutant has no communication, and what you can see is that the bacteria are colorless. They can't secrete the toxin, so they don't turn green. The right-hand test tube shows you wild-type Pseudomonas that we've added our inhibitor. And what I hope you can see is that the inhibitor greatly decreases the ability of Pseudomonas to secrete that green poison. So now we're in business. It looks like, at least in a lab, we can shut down biofilms and we can shut down toxin secretion. So what about in an infection? So in this experiment, you're looking at an animal model system that we have for Pseudomonas infection in the lab. And all we do is measure whether the animals are alive or dead. And so on the line that you're looking at, obviously, if we don't add Pseudomonas, the animals are perfectly fine. If we give a Pseudomonas infection, now what you can see is that all of the animals die within the first day after the infection starts. But if we do that, we give the Pseudomonas infection, and we give that inhibitor molecule that I showed you, what you can see with the third line is that we can greatly improve the outcome for the animal. So in fact, we think now that there must be merit to this idea of interfering with chemical communication, and that maybe this could form the foundation of a new type of therapeutic. And so what we're doing in the lab right now is we're taking the molecule that I showed you, and we have to make it more medicine-like. We have to build in potency, and we have to make that molecule safe. The second thing is that we got inspired by that biofilm experiment that I showed you, and we're working with engineers now to try to embed those inhibitor molecules into materials. And the idea is that maybe we could make infection-resistant catheters or stents or breathing tubes. And then finally, I'm just telling you one little vignette that's about Pseudomonas. We work on lots of globally important pathogens in my lab, and we're having similar success doing these kinds of strategies in other bacteria as well. And then to finish, I just want to show you the two students who did the work, Colleen O'Loughlin and Knut Drescher. They both work in the lab, and I'm lucky to get to work with them every day. Thanks for having me back.
That's so interesting.
So that's Bonnie Bassler and her work, and here we're able to, what's really cool about this particular talk, well, the reason I selected it is because it brings in what we learned in Unit 1 when we talk about biofilms and how we mentioned quorum sensing and how bacteria talk to each other. And now here in Unit 2, we can see how this is related to genetic or environmental influences on gene expression. We can see how quorum sensing is, we have a environmental signal, and the bacteria respond to that signal by activating certain genes for toxins or virulence. And then, of course, we can see the application of this research, how we can, how Bassler's team is trying to come up with new novel therapeutics. So I did have the honor about 2 years ago to meet Bonnie Bassler at a conference, and we had a great conversation. It's the second time actually that I've gotten to meet her. I met her when I was at Hopkins. So she really is an amazing scientist, and I love following her work. So Bonnie Bassler, if anyone wants to follow her stuff. So one of the ways that bacteria can control gene expression is through what's known as a two-component system. And this actually is unique to bacteria. We do not have this in eukaryotic organisms. And in this two-component signal, we have two proteins. And these are transmembrane proteins. One protein is called the sensor, and it's going to actually receive some type of environmental signal, whether it's a chemical signal. Let's say this is Bassler's quorum sensing and concentration dependent. So the chemical that's being released that says, hey, I'm a Pseudomonas, how many of you are out there? That chemical signal that's being released will start combining with, let me get my little pen here, we'll start combining with our sensor. And this is really called a sensor kinase. And a kinase is an enzyme that transfers phosphate groups. So our sensor protein here will receive this, bind to this environmental signal. Notice the signal is not brought into the cell. All this signal does is cause a conformational change in this protein that will hydrolyze ATP to release a phosphate group to make ADP. When it does that, that is an energy release. Remember, ATP is the ATP is the energy currency of the cell. Our second protein is the regulator or response regulator. So here is our response regulator. The phosphate group from this ATP gets bound to our response regulator. This response regulator binds to the operator region of a gene. This will activate RNA polymerase, which will then begin transcription of the gene, or be blocked from transcribing the gene. And then this will lose its phosphate group. The response regulator is now ready to receive a new one. And we, of course, have our sensor kinase here will eventually lose that phosphate group and go back to its original shape to start the process again. So we have a two-component system. We have one protein, our sensor, that's going to receive the environmental signal and phosphorylate the response regulator. The response regulator is going to tell the genetic material to either transcribe the gene or block transcription of the gene. Now, natural selection is another form of environmental influence. In natural selection, this is that kind of survival of the fittest, kind of Darwin's theory here. And here, survival of cells that are the best adapted to a particular environment is going to promote the increase in population of bacteria cells that are able to survive in those environments. So what I'm going to show you here is a quick video on this was a antibiotic resistance experiment that was done at Harvard Medical School, and in their bacteria lab, they took this giant it's called a mega plate, this huge plate, and they were looking at the actual evolution of a single strain of bacteria over time to different concentrations of the same antibiotics. So let's take a look at this. This is a natural evolution of a bacteria, of a single strain of bacteria to multiple concentrations of antibiotics.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So that bacteria was able to evolve resistance to a single antibiotic in at 1,000 times the normal medical, or the normal, what's the word I'm looking for? Trying to think of it here. Normal levels that you would give a patient. It's late and I'm trying to come up with this. That what would be considered normal levels in the system dosage. So 1,000 times a normal dose. And in 11 days, that bacteria is able to evolve resistance to it. Now, there are a couple of different ways that this occurs. One important type of natural selection or form of natural selection is what's referred to as antigenic variation. And this is really common actually in protozoans and some helminths. It's also seen in some bacteria. So in antigenic variation, what we see is a cell will alter its surface proteins and receptors. We see antigenic variation a lot in viruses, actually. So let's take COVID, for example. We're talking about COVID. COVID is very closely related to SARS and MERS, extremely closely related. They all fall in the same coronavirus family. And when we are hearing these reports about mutations, right, variations or substrains of COVID that are starting to pop up in different parts of the world, these are genetic mutations that could slightly alter those surface glycoproteins that are on the surface of COVID. And by altering those spikes, if they're just a little bit off, well, that is the antigenic marker for that virus. And our antibodies may not be able to recognize that viral spike, so our antibodies would then not necessarily be effective. This is why when these new variants are discovered, the first thing they start doing is testing antibodies, vaccine-induced antibodies, to make sure that the vaccine that is currently being circulated right now will still be effective against these different mutants or variations of the virus. And so far they have been, so that's a good thing. Another form of antigenic variation is phase variation. So instead of just altering a surface protein or receptor so the immune system can't recognize it, here in phase variation, genes get turned on and off at different levels. So a few examples of this we have here on the right. In antigenic variation here, I'm going to take these gray colored flagella here, and I'm going to change them to gray and black flagella. In phase variation, I have a genetic switch, and I'm just going to turn off the gene for gray and only express the gene for the black ones. So now the flagella are entirely black. In a phase variation on-off switch, right, because this is up here, phase variation anagenic switch, and a phase variation on-off switch, I'm just not going to produce any of the flagella, and so now I have no flagella. So I'm either going to alter the antigen, make it a combination of things, try and confuse the immune system. I can do an antigenic switch where I just change the antigen entirely, or I can do a phase variation on-off switch, I just get rid of the antigen entirely. So those are different forms of phase variation. Now, other types of gene regulation. We have global control, where we regulate a whole bunch of genes in one sitting. An example of this would be operons. Again, operons are unique to bacteria. They are where we have multiple genes all being regulated by a single regulatory region. This is an operon up here. Okay, so here we have an operon. Here are all of the genes that are associated with this operon. All of the genes within an operon are all going to encode for proteins that are associated with a similar function. We're only going to express, if we were a bacteria cell, we would only express all 4 of those genes if we are going to carry out that particular function. So in this case, with an operon, I only have to regulate this one region in order to turn on or off all 4 of these genes. This is a big space saver, a big time saver, and it's a much easier form of gene regulation for a cell as small as a bacteria. Then we have what are called regulons. In regulons, we have a whole bunch of operons that are all being controlled by its own single regulatory region. So I have here I have multiple operons, right? So I have multiple genes against galactose, maltose, lactose, arabinose. These are all probably enzymes for all of these produce proteins necessary for the metabolism of all these different sugars. But if none of these sugars are present, I can simply regulate all of them with what's called a second messenger or cyclic AMP. So here I can regulate the CAP protein without going too crazy. This catabolite activator protein, this protein here is used to regulate all, here it is, to regulate all of these genes. Okay, so I use the same protein to regulate, to say activate all of these genes. If I want to turn them off, I have high cyclic AMP levels. They're going to attach to all of the CAP proteins, and all of those CAP proteins then bind to the promoter regions of a whole bunch of operons, and now all of these operons are all turned off by simply regulating the single CAP protein. So I can regulate multiple operons, or I can just regulate a single operon. Many times enzymes get grouped by what type of mechanism they regulate. For example, we have constitutive regulation, inducible, and repressible. So in constitutive, these are genes that are constantly being transcribed and translated. These are genes that are responsible for the proteins necessary for everyday life. Let's take metabolism, for example. Metabolism is run, all of our metabolic pathways are run and function due to the creation of all these enzymes. Those enzymes are from structural genes. The instructions for those are from genes. So a constitutive gene is one that we're going to produce on a regular basis all of the time. Central metabolism, again, it's this constant synthesis. So sometimes enzymes are considered constitutive enzymes because they're part of that constant synthesis, particularly central or what we call primary metabolism. Inducible genes are the Lac Operon. So inducible genes are genes that when we need them, there'll be some type of environmental signal that says, hey, we need the products of those genes. And we'll go ahead and we can induce the transcription and translation of that set of genes. And then repressible genes are ones that are routinely synthesized, but sometimes we don't need them, so we're going to turn them off. An example of that in bacteria would be what's called the trip operon. It's part of a biosynthetic pathway. This operon here is responsible for producing the proteins and enzymes necessary for the synthesis of tryptophan, an amino acid that's used in protein construction. When tryptophan levels are high enough and the cell does not need to produce any more tryptophan, it will actually turn off that operon. And so that operon would be repressed, whereas another operon would only, that's an operon that's on all the time and turned off when we don't need it. The other is one that's turned off all of the time and then induced or turned on when we need it. So the enzymes that come from these different groups they're grouped by their regulatory mechanism. Is it a constitutive enzyme? Is it an inducible? Is it repressible? Other mechanisms of control include transcription. So now we're looking at the transcriptional level. At this level, we can, for bacteria, we can regulate the sigma factor. Remember, the sigma factor is part of what RNA polymerase is going to recognize. So if we bind or block the sigma factor, then RNA polymerase is not going to be able to recognize its promoter. Some bacteria may use an alternative sigma factor. This will allow for recognition of different types of genes. So this gene uses sigma factor A, and this one uses sigma factor B. So these are sometimes called special occasion genes, and these alternative sigma factors are unique and specific to those special occasion genes. Bacteria also, as well as our own cells, will produce repressors. In repressors, there are two different types: induction repressors and repression repressors. In induction, this is just like an inducible gene. Induction, we're going to synthesize, an induction repressor will be synthesized in order to prevent transcription from occurring, so the gene is always turned off. That would be like the Lac repressor. In repression, a repressor repression, we're going to synthesize the repressor and allow transcription because it comes with a co-repressor. A co-repressor is going to bind, causing a change in shape to the repressor so that the gene will be either turned on or off. Then we have activators. Activators turn stuff on. So repressors are negative control and activators are positive control. So if you have something that's under negative control, the gene's going to be off. If it's under positive control, the gene is going to be turned on. So activators will turn genes on, repressors will turn genes off. So here we have our different types of repressors. This is from your textbook. Here with induction, in the process of induction, RNA polymerase is bound and ready to go, but there's a repressor that's blocking. It looks like it's sitting on the operator region. So this is your typical like Lac Operon. We can induce it by introducing an inducer. The inducer binds to the repressor, causing a change in shape. The repressor cannot bind, and so transcription will occur. In part B repression, our repressor is unable to bind, so we add a co-repressor molecule that when it binds also changes the shape of the repressor, but instead of allowing for transcription, instead of removing the repressor, it actually causes the repressor to bind. And so when we have an inducer, we're going to turn our gene on, and when we have a co-repressor, we're going to turn our gene off. Here is our activator. In an activator, normally RNA polymerase would not be able to be activated if our activator, such as a sigma factor, doesn't bind to the promoter region or in the operator region. So we have this, what's called the activator binding site, this area within the regulatory region in front of a gene in which this activator or repressor or induction, one of these types of proteins are going to bind, and these are sometimes called transcription factors. So this activator here, in order for RNA polymerase to be activated and be able to attach to the DNA and transcribe the gene, this activator has to be attached. So an inducer molecule will bind to the activator, changing the shape of the activator, causing it to bind to the activator binding site. RNA polymerase says, oh, there you are. I have permission. All right, cool. Thank you. And then RNA polymerase is able to carry out transcription. But without that activator on there, RNA polymerase won't be able to carry out the transcription. So we talked about the Lac Operon, and the Lac Operon is an inducible operon. and it is used to encode for enzymes that are responsible for the breakdown of lactose. There are three genes, structural genes found in the Lac Operon. The first is LAC-Z, which encodes for beta-galactosidase. Beta-galactosidase will break a lactose molecule in half. Then we have the LAC-Y gene, which is permease. This is the lactose pump that's found in the membrane, and it's going to bring lactose into the cell so that the beta-galactosidase can break it into glucose and galactose. And then the last is transacetylase, and transacetylase is actually part of central metabolism. It speeds up central metabolism. It's not so involved in lactose metabolism per se. It's not necessary for lactose metabolism. It just speeds up central metabolic processes. So we're going to be concerned mostly with LAC-Z and LAC-Y. Now, the Lac Operon is an inducible gene, which means it's an inducible operon. So it is normally under negative control. If I'm in a normal glucose environment, I have no reason to produce these enzymes because there's no lactose present. So in this case, the gene is inducible. It is normally turned off. We're only going to turn it on if lactose becomes present. Now, there is some what we call constitutive transcription. Every once in a while, the repressor will fall off. We'll produce a couple of copies of these genes, and this will keep our lactose pump in the cell membrane so that if lactose does become available at a later time, we'll be able to sense it. If we don't ever produce any lactose pump, it doesn't matter how much lactose is in the exterior environment, right, outside of the cell. If I can't get it inside of the cell to be broken down and used for energy, there's no point. I'm not going to be able to turn on my gene. I'm not going to be able to metabolize it. So let's take a look first at the actual structure of the Lac Operon itself. Over here, all the way upstream outside of the operon, we have a gene here called the LAC-I gene, and it encodes for the Lac repressor. Then we move a little downstream. There's some space, and then we have a region called a promoter. Now, in the regulatory region, right here, the zones where proteins bind, this is the regulatory region. And in that regulatory region is the promoter and operator. The promoter is basically where your sigma factors and your activators are going to bind, and where RNA polymerase, all of these transcription factors all come together and say, hey, RNA polymerase, let's get going, let's get activated. The operator is where our Lac repressor binds. And when the Lac repressor, which is not regulated by the same genes, the Lac repressor is constitutive. So it's always being transcribed and translated. And the Lac repressor will bind to the operator. And the only way to remove the Lac repressor from the operator is to bind the actual lactose molecule. Its inducer is lactose. So if lactose comes into the cell, it will bind to the Lac repressor that's sitting on the operator and cause that Lac repressor to fall off. If the Lac repressor falls off, then RNA polymerase now has the freedom to move forward and carry out transcription. But the Lac Operon, it's not being supported. We're not able to play this movie right now. So I can't play this movie right now. Hopefully in your ebook you should have access to it. You definitely want to watch this movie. It's a great explanation of the Lac Operon. In the Lac Operon, let me see if I have another. Nope, I'm going to go back here to here. So when my Lac repressor is sitting on here, in order to remove the Lac repressor, I have to add a bunch of lactose to this guy. And this is going to cause the Lac repressor to fall off. Now, RNA polymerase wants to move forward and transcribe, but RNA polymerase has to be activated. And the CAP, or catabolite activator, can only bind, right? It can only bind if cyclic AMP is bound to it. So in order for me to transcribe and translate my Lac Operon, I need a couple of things. I need my lactose to be able to be brought into my cell, and the lactose molecules bind to the Lac repressor that's sitting on the operator. I also need cyclic AMP. And the cyclic AMP is necessary to get my catabolite activator protein, to get my activator protein to bind to the promoter in order to turn on RNA polymerase and push it forward so that transcription can occur. So I have two layers of regulation here. I need my lactose layer with my lac repressor, but I also need cyclic AMP. And what makes this really tricky is that cyclic AMP levels are controlled by glucose metabolism. So in an environment where glucose is in a really high concentration and lactose is present, my gene is going to be turned off, because I might be able to activate, I might be able to remove the operator because lactose is present, but I cannot activate transcription because I don't have enough cyclic AMP. When glucose is being metabolized, all of the cell's cyclic AMP is going to be used up in glucose metabolism. So the cyclic AMP levels are really low, and the operon cannot be transcribed. So glucose regulates the cyclic AMP levels, and lactose regulates the repressor. Cyclic AMP regulates at the promoter, lactose regulates at the operator, and that cyclic AMP is necessary for RNA polymerase activation, and that cyclic AMP level, those concentrations, are controlled by glucose metabolism. Okay, this movie is not working. I know that now. There we go. Now, this is what's called the CCR, carbon catabolite repressor, and these are some examples of glucose and lactose metabolism. When sugars are getting broken down, we can measure this through time of incubation. So they're looking at how quickly, in this graph, they're looking at how quickly cells grew on glucose and then when they grew on lactose. And what happens here, you can see they're pretty much the same rate. Lactose growth was just slightly slower, but glucose and lactose were added to the media. We went through our initial growth phase, and now we start in this logarithmic phase. And in the logarithmic phase, these guys are using glucose for metabolism. So growth is very quickly. They don't have to wait for anything. But right about here, all of the glucose in the media gets used up, and the bacteria no longer have glucose for metabolism. For a short period of time, we have no growth. And what's happening here in this period of time is all of this. The cell is taking all of that lactose, it's bringing in that lactose, going, hey, you know what? I may not have glucose, but I've got lactose, so let's get this Lac Operon going so we can start using lactose for an energy source. It does not want to, there we go. Once it takes a few minutes to get through this, okay, we're going to move from glucose metabolism and start inducing our Lac Operon. Now the Lac Operon is activated, and we start seeing growth on lactose. Lactose is now able to be broken down to provide the power necessary for microbial growth, and eventually, and that growth will continue until eventually the lactose also gets used up, and then you can see here our growth levels off. This type of growth where we have this kind of hiccup or this little stall in the middle, this is referred to as dioxic growth because we're looking at two different sugars here. So here are some examples from an actual research paper. This is E. coli being grown on different types of sugars. Now you can see here there's a slight dioxic growth, very little. Same thing with fructose. By the way, these are fructose is a monomer, right? Those are monomers, galactose too. But in the bottom here, we have, we're starting out with a combination of sugars. So glucose is a monosaccharide, and xylose is a disaccharide. Glucose monosaccharide, arabinose disaccharide. Glucose monosaccharide, rhamnose is, I believe, a disaccharide. And you can see here we have this dioxic growth. So growth was going really well, and then it broke up. It stopped for a while we saw some kind of change in the gene expression, and then it started up again. So again, here is that Lac Operon. We're going to have our repressor bind to the operator. The promoter is going to need the cap with cyclic AMP attached to it. RNA polymerase to be activated in order to move forward. It's only going to transcribe these genes if both the repressor is gone, lactose is present, and cyclic AMP levels are low, glucose is absent. Now, in eukaryotic gene expression, we have a couple of different forms of control mechanisms. I'm not going to go into great detail in these. Just some of them are pretty straightforward. The first is what's called epigenetics. In epigenetics, we modify the chromosome. So we can like tighten up parts of the chromosome or loosen up parts of the chromosome to either prevent or enable transcription and translation. Of course, we could regulate the actual initiation of transcription, similar to what we do with glucose levels, cyclic AMP, and RNA polymerase. We can alter the amount of pre-mRNA. We can alter the pre-mRNA that's found in the nucleus. So our eukaryotic mRNA has to be processed before it can be read by ribosomes and translated. So this occurs in the nucleus. We can alter that pre-mRNA so it can never leave the nucleus and won't be transcribed. We'll reduce that protein production. We can also alter post-mRNA. This is done in the cytoplasm. This is done by creating complementary RNAs that might bind to it to make the RNA double-stranded so ribosomes cannot read it. There could be ribozymes, which are RNA molecules with enzyme activities that may break mRNA molecules into a multitude of pieces so that the ribosomes can't read them. RNA interference is a pretty common form of RNA regulation what we call post-transcriptional regulation, where single-stranded RNA, now remember, messenger RNA is single-stranded, but it's still bases. So if I create a complementary strand to that single-stranded mRNA, even just regions of it, a few regions of it, the ribosome is not going to be able to transcribe it. So what's called RNAi, or RNA interference. RNA interference was discovered by a scientist, by the way, by the name of Dale Emeagwali. She's going to be featured in our Black History Month announcements. And then finally, there's the process of genomics. Now, in genomics, we can sequence a genome, and this is just more of a biotech type of thing. But in DNA sequencing, we can take genomics and we can do what's called a blast. And in the blast, this is done through the NCBI, and this is the National Center for Biotechnology Information. This is where throughout the world we can share gene sequences. And if we know gene sequences, then we can start learning about the behaviors of different regulatory proteins. We use computers to detect what are called protein coding regions, and they're called open reading frames. So they search for 300 or more base pairs that have both a start and a stop codon. That gives us a general idea that that's a region in which a ribosome might actually bind. So in genomics, we're just simply sequencing the genome of organisms and entering that into a bioinformatics database so we can then search it and look for different patterns. In metagenomics, we study the genomes of an entire population, not just a single species. So in this case, we might look at the genome of several bacteria in soil or different microorganisms found in water. We can even look at the gene expression of different genes within the same genome in a population as well. This is usually done to see relatedness between microorganisms that are growing in similar environments. So that is it for part 3. The last couple slides there were just kind of introducing you to the idea of biotechnology, and we move into that in the next couple chapters. So I hope you enjoyed chapter 7. It was really fascinating stuff. It's kind of cool. And I will see you in class. Have a good night. Did I just do that whole thing and not record it? Oh my God. Seriously? Seriously? No, I got it. It's still recording, so how do I stop?